Look at that, a pre-hushed audience. We can go ahead and kick off with housekeeping. So kia ora, welcome. I'm Abby Albright. I'm a business manager with the Bioeconomy Science Institute. Thanks for carving time out of your busy schedules for today's Research Snapshot. We've got 3 updates today, all from MB programs that relate back to the wine industry. Just a bit of housekeeping at the front. I think probably most people are familiar with the facility, but in the event of an emergency, the mustering point is just out the car park where most of you will have parked your cars. In the event of an earthquake, stop, drop, and cover. Do mind the heavy clay tiles that surround the courtyard after the shaking stops if we evacuate. The loo blocks, there's a couple of toilets just out and to the right, and then catty corner to where we're at in the courtyard is the New Zealand Wine Centre and another set of bathrooms there. So thanks very much for coming. Our first presenter is Michelle Thompson. She leads out an MB Smart idea that has its origins back from when there was a person out wandering the vines in the National Vine Collection and observed in an otherwise high incidence of mealybugs. There were a few vines that didn't look very tasty to those bugs, and that was the genesis of the idea. And now 3 years on, with a 1-year extension to the Smart idea, Michelle is leading out that program and making us all look bad with her amazing multitasking, because at the same time as doing her doctoral studies, in addition to leading out the program. So I will let her give you an update on what they've found out so far and the direction of the program. So welcome, Michelle.
Right, thank you for that wonderful introduction. That makes it easy to start with what I'm going to be talking about today. So I'm a co-leader of the program, and as Abby's just said, I'm also a PhD student and I'm studying at Lincoln University. So today I'll be talking about our work investigating grapevine metabolites as potential feeding deterrents for Citrophilus mealybugs, which are pictured on the front slide here. So on the left here is a female Citrophilus mealybug, and at only about 2 millimetres long, it's tiny, but it's a big threat to the New Zealand wine industry. And that's because during feeding, these mealybugs can transmit grapevine leaf roll virus. And grapevine leaf roll disease, which is caused by the virus, can have negative impacts on productivity, yield, and grape quality. So it's a big deal for the industry. Not only that, but during feeding, the mealybugs also produce honeydew, and that can have negative impacts on grape quality as well, due to the production of black sooty mould. So as Abby said, what we know is that mealybugs prefer feeding on some grapevines over others. And so we want to know what drives those preferences, and can we identify what makes them prefer some grapevines and not want to feed on others in particular. And if we can identify in particular metabolites that drive those feeding preferences, that could support breeding programs to maximise natural resistance to these mealybugs. So I want to start by talking about some of the foundation work, and the 3 people pictured in the top right of the slide were involved in the work before the MB program started. And what they did is they established a way to assess mealybug feeding preference on different varieties. And they did this by looking at establishment, development, and survival of mealybugs on leaves of the varieties pictured in this table. So these ones here. And they then ranked the mealybug feeding preference on these different varieties. And what you can see in the table is that Ripara Gloire, for example, which is a rootstock, had a high mealybug feeding preference, and Schwartzman, another rootstock variety, had low. And here a low feeding preference means that the mealybugs would avoid feeding on these plants when provided with a choice, and when given no choice but to feed on them, they would develop more slowly, progress through their life cycle more slowly, and it negatively impacted their survival as well. So what these researchers did is they then did metabolic profiling of the leaves of these varieties, And that identified a class of compounds called the elagitannins as a leading candidate associated with mealybug feeding preference. And the MB program is centred around these elagitannins. So very loosely speaking, the program is centred around kind of 3 key questions. And rather than try and talk about every little thing that we're doing, I'm going to talk about the parts listed here in italics. So just to give you a snapshot of some of what we've done. So firstly, how much variation is there in elagitannin content? And today I'll talk about how we've screened the leaf elagitannin content across grapevine germplasm. Then where do elagitannins accumulate in grafted grapevines? So we'll talk about elagitannin distribution across tissues and also looking at different graft combinations. And do elagitannins affect mealybug feeding? And as Abby said, we're very fortunate. We've received an additional year of funding for this program, and that's allowed us to extend our work in this area in particular. And I'll talk today about our most recent work where we've developed a controlled feeding system and a way to directly assess the effects of elagitannins on mealybugs. So first I'll talk about leaf elagitannins across grapevine germplasm. So what we did is we measured the total leaf elagitannin content in 6 Zion varieties and 28 rootstock varieties. And you can see the varieties listed down the left here. Sorry, they're a little bit hard to read on the screen. Leaves were collected in March and in May of the same growing season. And what this figure shows is the average leaf elagitannin content relative to the mean. So the bars shown in green, these are varieties that have a higher than average leaf elagitannin content, and the varieties shown in pink have a lower than average leaf elagitannin content. And here I've highlighted 3 varieties that we're quite interested in, we continued looking at throughout this project, which is Schwartzman, which we know has low mealybug feeding preference and relatively high leaf elagitannin content. Riparia gloire, which the mealybugs really like feeding on those leaves of this variety, and it has a low leaf elagitannin content. And we also selected Sauvignon Blanc, which produces some elagitannins in the leaves, but very low elagitannin content. And that's similar to a lot of Zion varieties. So you can see Cabernet Sauvignon and Syrah, for example, at the bottom here. And then what we did is we continued with the selected varieties, so Sauvignon Blanc, Riparia gloire and Schwartzman, and we then set up potted vine trials to answer questions about the elagitannin chemistry in different parts of the plant and the effect of grafting. So our research questions here, where do elagitannins accumulate within grafted grapevines? And does grafting affect elagitannin content in the scion? So to answer this, we set up potted vine trials in the shade house. This is the shade house trial photo here, and in the glass house. And we collected leaves, petioles and roots at 2 different time points, and this was conducted over 2 years as well, to look at elagitannin content across the plant and the impact of grafting. So we used various grafted and ungrafted combinations of these 3 varieties. And what I'm presenting here is just a small amount of the data, just to give you an idea of the scale of the thing. In the first year of the shade house trial, there was over 1,000 samples, so it's a lot of data that we have here. What we have is trend line plots, is what these are, and they show accumulation of all of the identified elagitannins in the different tissue types. And each line on one of these plots is an individual elagitannin compound. And it's hard to look at it like this, but what you can see is that you can sort of follow along a line and see what the general trends are. It is, however, a whole lot easier to look at it like this, which is a single elagitannin compound. So this is a single elagitannin compound across the 3 different tissue types, so leaves, petioles and roots. And I should say we selected these tissue types because we know that Citrophilus mealybugs will feed on all 3 of these tissue types in a grapevine. And then we looked at the abundance of this elagitannin in the different plant types. And so you can see here we've got Riparia gloire, there's basically none of this elagitannin in the leaves. And Sauvignon Blanc, which is the 4 middle bars here, there is some, but a relatively small amount. And then in the leaves of Schwartzman, we have a really high amount of this allogentannin. And that's consistent with what we've seen previously and with our feeding assay data showing that the mealybugs really, really don't like feeding on the leaves of Schwartzman. Importantly here, we don't see an impact of grafting on the allogentannin content in the scion. So these bars here just show in a visual way the different grafting combinations. We have Sauvignon Blanc on its own roots, Sauvignon Blanc grafted to Riparia gloire, Sauvignon Blanc grafted to itself, and Sauvignon Blanc grafted to Schwartzman, and the allogentannin content in the scion stays relatively stable. So importantly, this allowed us to set up a robust analytical method where we could analyse individual allogentannins, and we can now look at them in different tissue types, different parts of the year. There's a lot that we can analyse here. So I'll now move on to talking about the next part of our work, which is looking at the impacts of allogentannins on mealybug feeding directly. So what we know so far is that grapevine varieties differ in their allogentannin content. The allogentannin profiles differ among the different plant tissues. That higher allogentannin content is associated with reduced mealybug performance. And we know from other published studies looking at allogentannins that they can act as feeding deterrents in other plant herbivore systems. So they're kind of a known defence compound. Our next question is, do mealybugs directly influence mealybug feeding behaviour? And the key word there is directly, because so far we have a really strong association and we're wanting to measure impacts directly. So to do this, we needed to develop a controlled system where we can test the impacts of allogentannins in isolation. And we will be using an artificial diet to do that, and I'll talk about our development of this system today. So an artificial diet allows us to control the allogentannin concentration, so we can add different concentrations of the compounds to the diet, and it isolates the effects of allogentannins from other factors that may influence mealybug feeding. For example, the plant tissue type, the physical traits of those tissues, other secondary metabolites that could potentially play a role, leaf wax chemistry, and baseline leaf nutrients. And we can use an artificial diet to test whether allogentannins directly influence mealybug feeding behaviour. So the flowchart on the right here shows basically what we intend to do. We'll extract the candidate grape metabolites, allogentannins in our case, from grapevine leaves, add them to an artificial diet, expose the mealybugs to those compounds, and measure their feeding and performance in response and see if we see a change. So I'd like to acknowledge here Olalee Shenadeh, who's just finished her internship. She worked with me in developing the artificial diet that I'll be presenting today. And she's from Institut Agro-Dijon in France. What we did is we developed an insect feeding cage, and the cage design is inspired by similar membrane feeding systems designed for aphids. And so they have a similar feeding mechanism to mealybugs, so we kind of based our design on that. So basically it's two 3D printed rings, one on top of the other, and we stretch really, really thin membrane, in this case made of parafilm, over the end of each of those 3D printed rings. They're then sandwiched together with the artificial diet in between. And what that allows the insects to do is to insert their needle-like mouth parts through the membrane to access the liquid diet in between. And importantly, this is the mealybugs in one of our cage setups. And what you'll see is that there's also wrinkles in the parafilm. I won't present the data today, but we did look at this. Mealybugs really, really like to settle next to something that has a certain physical shape. And so they really like either the edges of the cage or wrinkles in the membrane. Whereas an aphid will probe in a place that's smooth and flat, mealybugs do not like that. So that was an important feature of our cage. So one of the things you'll notice is that our artificial dye is bright red. And the reason for that is that we've added a red dye to confirm that the mealybugs are ingesting the diet. And this is really cool because we can see physical evidence that they're ingesting the diet. So at the end of a feeding experiment, the mealybugs are gently removed from the cage. They're frozen. We have to wash them to gently remove the wax from their surface so that we can see inside the insect. And on the right here are some examples of what we see inside the insect. So at the top, this insect has no evidence of any dye inside it, and so we would say that there's no evidence of ingestion in that insect. In the middle is an example of where you can see the dye just in one particular location. This is very likely to be what's called the dilated sac stomach in the insect, so this is their guts. So the fact that we can see the dye inside the gut is good evidence that they did ingest it. And in this insect, we get more extensive dye distribution. So the dye has made its way through the body of the insect. Importantly, we do also confirm that the dye is in the gut of this insect as well. So this is strong evidence of ingestion. So now we have a metric that we can use to determine that the mealybugs are feeding on our diet and confirm that they are. So one of the things we did is look at different published diet formulations to make sure that we could find one that our mealybugs will like to live on. So we measured survival on a bunch of different diets. We used 3 published formulations, A, AP3 and D and M, both without the red dye and with the red dye. And what we found is that basically survival of these insects over a 4-day period on our diets was actually really very good. So this is 95% here. So you've got basically more than 95% survival of all insects over a 4-day period on all of the diets. We then looked at settling. Now settling is basically we determine whether the insects have their mouth parts inserted into the membrane and are accessing the diet at the time of recording. So this is after 4 days. And so we found that settling was best on this diet, AP3 plus red, and so we decided to proceed with that diet. We did also measure red dye uptake. I just can't present all of our results. And so the other thing I will say is that we wanted to make sure that the addition of the red dye didn't negatively impact our insects. And so we were able to show that. It is a non-toxic dye and it's something that has been used with insects before, but we wanted to make sure that Citrophilus mealybugs are okay with it too. And it certainly didn't impact the survival of the insects. And in fact, the addition of the red dye improved settling. So we then wanted to see whether our AP3 plus red dye would support prolonged exposure. So could we run our experiments over longer than 4 days to see how the insects performed over a longer time period? So we ran our experiments for 20 days. And what we found is that basically survival of these insects up until about 12 days was really, really good. So well over 95%. And then we saw a decline beyond that, to the point that there were very few insects alive at 20 days. We also looked at red dye uptake. So this graph is all insects that we confirmed dye ingestion because they had the red dye in the guts of the insect at the time that we recorded. So over a number of over up until about 18 days, you can see the red dye uptake in the insects is relatively constant, and then it does drop down at the 20-day point. So what that tells us is that for maybe up to 12 days, our insects are surviving and ingesting on our selected diet. And we also recorded molting and egg laying of these insects, showing that they developed in a relatively normal way on our diet. And so we now have this practical platform that we can use for testing the effects of elagitannins. So next steps that we haven't done yet, we'll incorporate elagitannins into the artificial diet, test the effects of those elagitannins on the mealybugs, and importantly, we also think that the artificial diets are going to be compatible with EPG measurements. So in other parts of the program, we've developed electropenetrography, or EPG, as a system for measuring real-time mealybug feeding behaviours. And this is just a picture of a mealybug attached to an electrode. And so we can measure the real-time feeding behaviours of these bugs, and we think we can do it on the artificial diet as well. So longer-term applications, we could even think beyond elagitannins. Obviously, the target of our program is elagitannins, but it could be possible to use the same systems that we've developed to test other grape metabolites that have potential to deter feeding. And any information about metabolites that deter mealybug feeding could support future grapevine breeding decisions. So I'd like to finish up by acknowledging my PhD supervisors. So this is Amber Parker, Philippa Barrell and Olaf Szaleski. And they also are part of the MBIE program research team, and the other members of the team are listed here. And there's also many contributors that have been involved either previously in the program or still involved in helping out with the expertise and things. And in particular, Ola Lee, who spent time working with literal thousands of mealybugs to set up the artificial diet. I'd like to acknowledge and thank MBIE for funding the program, the Bioeconomy Science Institute where I work, Lincoln University where I'm a student, and the Institut Agro-Dijon, which is where Ola Lee's from. That's the end of my talk. Thank you.
Any questions for Michelle? While you're cooking them up, just wait for me to bring you a microphone, which looks like a delightful little puffball for you to speak in. You may have noticed that today is our inaugural session recording these for those of you who are unable to attend in person, so that you can find them archived on the MRC's website.
That's very impressive, Michelle. Thank you. Schwartzman had the high larger tannins in the leaves. As a rootstock, is it going to have the same effect?
So one of the things we've looked at is the levels of larger tannins in the roots. And maybe the plot doesn't show it because the y-axis were all the same when we compared the different tissue types. But what we saw is that the levels of larger tannins in the roots of Schwartzman were elevated above, say, Sauvignon Blanc. So we do plan to look at the root chemistry as well, but I don't have a lot to share on that today.
Sort of along the, really cool work, but along the same lines of David's question, you said that you guys noticed there was less feeding on Schwartzman and that was sort of the origin of this. Have you seen less feeding on the roots of Schwartzman versus Sav or RG in the trial, or was it just merely foliar feeding that you saw differences?
All of the feeding behaviour studies are all done on leaves because we're trying to look at the larger tannins themselves as a candidate. However, again, we have the chemistry data on the roots. We haven't performed any mealybug feeding studies on the roots. As part of the foundation work, Mano, who had her picture on one of the slides, tried very hard to do mealybug feeding assessments on the roots, and it proved very, very difficult. So it's not something that is part of this funded program where the focus is on the larger tannins as a candidate compound, but it's something that I'd be really interested in the future.
Thanks, Michelle. Great work. Can you be confident that the work that you've done on the Citrophilus species is comparable with the Longospinus that we basically see around here?
Yeah, so part of the reason that Citrophilus is chosen is because we know that Citrophilus feeds on the roots. So that's why it was selected for this project. There has been work done, so particularly EPG work done on Longospinus mealybugs, and the feeding behaviour of Longospinus and Citrophilus are really very similar in those feeding assessments. I think it's something that we could definitely look into whether Longospinus mealybugs responded in the same way to large tannins, for example.
Thank you.
Thanks, Michelle. You mentioned your model system could be used for other metabolites. Excuse me. There's a bit of talk out in the industry around nitrogen and amino acid levels in sap. Firstly, do you know if there's any evidence that kind of proves those hypotheses? And secondly, presumably your system could test different amino acids, for example, in the feed.
Yeah, so the diet is essentially attempting to mimic sap. So it contains particular quantities of amino acids that has metal salts and vitamins. And so you could make a different diet where you tweaked the quantities of certain amino acids, or if you had something that you wanted to test in this diet by adding it to the diet, there's no reason you couldn't do that. So basically what we have now is a platform where if someone came to us with something else that they were really interested in testing, that would be possible. But certainly tweaking the amino acids, because they're added one by one to the diet, so the diet has about 40 different components, it's something that we could definitely tweak.
Any other last questions for Michelle? Paul.
Even though it's too early, do you have any clue what the larger tenant is doing to Mealybug?
Sure. So in one part of the program that I didn't talk about today, we looked at what the potential mode of action might be. And so there are sort of 3 possible modes of action for tannins as defense compounds in general. And we looked at maybe one in particular, which is kind of an oxidative activity kind of mode of action. In our studies, we weren't able to see that the larger tannins from grapevines necessarily had an oxidative activity above and beyond other tannins. What I will say is that that's the most likely way that they will impact the insects, is that they ingest them and then it basically is making oxidative damage in their gut, and then they go, do you know what, this is really feels really bad and I don't want to eat this anymore. That's the most likely way that they'll have an impact.
So do you see anywhere in the future using that chemical as a control method in general, maybe spraying it, or how do you see an application in the future using that compound to control mealybug?
Yeah, it's a really good question. So basically we would be looking at ways to breed grapevines with elevated levels of these compounds, because it's not something that we can apply, it's something that the insects need to ingest, and they ingest from the phloem and the xylem of grapevines. So it needs to be something in the grapevines, and we're looking at natural resistance.
Super. Thank you, Michelle. Really exciting work, and thank you for taking the time to drive down. Okay, bear with for 1 sec while we switch programs. Super. So moving from one form of natural defense to another, the next speaker we have with us is the Justine Larrouy, who's leading a program looking at vines that have shown resilience to trunk disease infection and how that relates back to their microbiome. So thanks very much, Justine. Welcome.
Thank you. Oh, yes. Oh, yes, I can. Hopefully. Okay. Thank you. So, as Abby says, this is a Smart ID MBI-funded project of a Microbiome-Engineered Grapevine. It's a 3-year project. We are on nearly end of the 2nd year. And this is led by the Bioeconomy Science Institute in collaboration with Lincoln University and Bragato Research Institute. This is, I'm co-leading this project with Hayley Ridgway, but you have a big team behind it. We have Simon Billman, Dion Mundy, Ferlanda, Nito Jacobo, Sandy Kennan, Damola Adagero, and Abby, of course. But also we have Ian Jones from Lincoln University, Beatrice Fulton, who is a PhD student funded by this project, and Banu Pratap Vanga from Bragato Research Institute. So what is all about? I'm sure you have heard of, but like human plants are associated with a diverse community of microbes, and we term this the plant microbiome. I have multiple examples in the literature showing how microbiome impact key traits in plants, such as growth and development, disease resistance, but also production of secondary metabolites. And we use these microbes already when we use biocontrol, but the future vision is to look at the community of microbes that is present and associated with the plant, and to use this as a functional gain for the plant, because this microbial community are also additional genetic powers that the plant can tap into. But the science behind it is quite complex and quite novel as well, because we need to determine who are the freeloader, they're just on the plant and don't do really nothing for the plant, versus the ones that really will bring a gain. And there's also a community aspect of it. And so creating a plant with manipulated microbiome is really at the edge of what is possible at the moment. But we are trying, and this is a good tool. And I mean, grapevine trunk disease, it's a perfect disease to test this on, because grapevine trunk disease is caused by many fungal pathogens. The symptoms of this disease appear later in the life of the vine. Of course, Sauvignon Blanc is one of the most susceptible varieties, and across to New Zealand, but also globally. And at the moment, the tool we have against this disease are best practice. And in the current timeline, if a vine is infected, it will start showing symptoms later, start losing yield, and end up being replanting around 20 years old. Where if we had a tool against this disease, we could actually have vines that are producing until the end of a natural lifespan, which will be 30 years plus. So this project is actually built on previous work and premier work done by Damola at Agero. He was a PhD student at the time. And what he did is he took a small microbial community from disease escape vine. So what are disease escape vine? Dion worked a lot on this vine. Is in a vineyard where a lot of plants are infected by grapevine trunk disease showing symptoms or dead, you have this random plants that look healthy. And so we call them the disease escape vine. And so he drilled trunk and isolate small microbial community from this disease escape vine and transfer them into a young vine. And from this, he challenged this young vine with grapevine trunk pathogen and looked at disease progression. So he looked at the lesion measurement. And what he finds is that the vines that were inoculated with the microbial community had smaller lesions. So what did they really do? They changed really the scenario of not leaving a grapevine microbiome to be formed randomly, but as also shows that disease escape should be the norm and not the exception. So he published some of the data as well on this, and more are to come. So from this, we built this project where we want to do this work, but in a bigger setup, and to have a plant that will have manipulated the microbiome to augment the plant to be more resistant to grapevine trunk disease. And three things we aim to deliver with this project is specification on how to design this community, what technology we should use to infiltrate them into a young vine, but also a provisional operation plan to integrate this microbial community into current nursery procedure. So year one, we've spent the first year designing our microbial community. And this is complete, but it was not an easy thing to do. And I'm not showing all the results here. I'm just trying to show you how we did it. So we went data mining. We had so much microbiome data of disease versus disease escape plant. And we look at what bacteria were present in the disease escape plant versus disease plant. And in parallel, we use microbial isolation and functional testing. So as you can see on the graph on the left, if you just look at the yellow part of it, here in the disease escape plant, we were finding a lot of, so relatively in their microbiome, Pseudomonas bacteria was very abundant. So this was a perfect target for us. And at the same time, samples from this vine were taken to isolate bacteria and testing them. So here on the right, you have what we do to test if the bacteria will have an effect directly against GGT pathogen. And so here at 0, you have just the fungal pathogen growing on the plate without nothing. It's growing well, happily growing, but when we add bacteria, and this is different, we can rank them. When we have bacteria, the growth of the fungal pathogen is reduced. So this plus this give us a good indication that there will be good candidate for it. What we did is we used kind of a decision tree to know what are we accepted and what we rejected in our microbial communities. And we decided to made 3 microbial community. So we have the simplistic community with 7 bacteria and 2 fungi. And 7 bacteria are 7 Pseudomonas, but 7 different Pseudomonas. So it means that there are different species or strains that were taken from the same or different disease escape vine. And we have the medium complexity and the high complexity, so up to 27 different bacteria and fungi within our community. And we did a Russian doll approach where we have this 9 microbes are in this 18 microbe, and this 18 microbes are in this 27 microbes. So we're just adding more complex and diverse community. So in year 2, we needed first to infiltrate this community and follow them. And this can be quite tricky, and this is current work. So what we've done is we use vacuum pump infiltration technique. As you can see on the video, we take cane and we connect them to a vacuum pump. We just, in the liquid will be our microbial solution up to the 27 microbe, and we'll just infiltrate them. So it will be a 10-15 seconds per cane, and you can see it coming out on the other side. And after this cane are planted, and we grow them. We also trial a soaking method, just to put the cane into our microbial solution for a few days, just to see how it will work, and this is to link with a nursery procedure. We've done more work on the vacuum pump infiltration because at this point of the project, we want to make sure that we have the best method to follow this community and then they are infiltrated. But more results are to come on the soaking method. In this year and in this project, one big thing for us is to follow what we have put in. And so we need to develop method to track the microbial community. And for this, we have developed a non-GMO method for the 7 Pseudomonas. And we use this 7 that we can follow individually as a proxy for the different microbial community. But we're also developing a molecular method for the fungal member, and we're using microbiome analysis as well. So I have only results for the simplistic community at this point, more results to come, but 6 months post-infiltration, we are using 2 methods. So we are taking cane tissue for the plant we infiltrated, and we're plating them out to see if our bacteria are alive. And out of the 10 plant tested, 10 show our bacteria back alive. 3/4 out of the 7. But for us, it's very promising. And the ones that we don't find with this method doesn't mean it's not there. We might need to use another method to make sure that they're there. But also, and we were very surprised by how clear the microbiome data show us the same result. So on the infiltrated plant and light blue, these are our Pseudomonas bacteria versus our control plant. So we are going back to a GG escape microbiome pattern by re-infiltrating them. This work is heavily done and supported / I will say done by Beatrice Fulton, the PhD student in this project, because she will be working only on the simplistic community and trying to track this bacteria and yeast up to 24 months. And she's not only tracking them in time. She also is looking where are they. Are they only in the cane? Are they in the stem? Are they moving in the leaf? Are they in the roots? We know them at the moment. We find them in the cane. We don't find them in the leaf or in the stem, 6 months post-infiltration. But now we know that they're in the cane. Where's in the cane? Are they in the phloem? Are they in the xylem? All of this type of question. She's got a part of a PhD that will be that is very interesting and more linked to industry, where she actually collected cane sample throughout the nursery process to look at how the microbiome change at the different steps, to provide insight on when will be the best time for us to inoculate this cane with the microbial community. She also has another part of her work where she will try so she will infiltrate cane and graft. And she will just do this with Sauvignon Blanc, but she will see if this microbe could cross a graft union. And another part of her PhD, she doesn't have enough work. She will be also try to inoculate this simplistic community into the trunk of mature vine. We know that this is going to be more complex because these vines are mature, they have a mature microbiome, so trying to put new microbe in it will be more difficult. The plant might have more difficulty to uptake this microbe rather than a young plant. And so she will be using syringe, and we just received Dion just bring us some mature vine last week, so she will be starting on this in a couple of months. Finally, what are we going to do on the last year? So on the last year of this project, we want to test if our microbial community work against grapevine trunk disease. And for this, we are going to challenge our plants with some grapevine trunk disease pathogen, looking at disease progression. So hopefully this is what we want to see in year 3, and see what we have against the simplistic, the medium or the high complexity, but also looking at functionality. So we will try to start uncovering if our microbial community help the plant against grapevine trunk disease, how does it work? So we'll be looking at the gene expression of the plant to understand if some defense mechanisms are triggered in the plant with a microbial community versus the plant without. And that's it for me. Thank you.
Questions for Justine?
Thank you for your presentation. That was well done. My question is about the microbial community on your disease escape vines. Mm-hmm. So where was that composition or that microbial community exactly the same across different disease escape vines?
No. And this is a good question. It's actually a bit more complex because we have few in different vineyards. So this also, we had to look globally, and some actors were the same, like some genera, for instance, of bacteria were the same, but some were not. So what we did is we looked in this vineyard, what's the profile and what is different to the vines that are next to it, because we take into account as well the soils they are in, the environment they are in, and this could change or affect the microbiome of the plant itself. So we went and looked at all the data and we have and say, okay, in this disease escape, this is the profile versus the plant around them. This is the profile that we have. And this is microbes and we isolated from it, because this is quite of the difficult part as well. We need to make sure that we have the isolate, then correlate to the microbe. And so we're like, these are the best from this vineyard, these are the best from this vineyard, and put them all together. So, yeah. It would have been easier if they would have had all the same microbiome profile. We can see some key actors, but yeah, have the field and environment variability.
So how do you know that the composition or the microbial community that you are composing here is the best one?
I don't know.
Okay.
I'm hoping so. What I will say, we did a simplistic because there's no low diversity. But when we add some diversity of microbe, we are hoping then some will take over as well, and some may not act directly against the pathogen, but just help, again, like environmental stress. So they may not directly impact against the disease, but they may help the other to do so. So, yeah, it's a good question. I think when we will see our 3 community and how they, with a challenge, we will see. We might have the number 1, the sample community, and have same results than the high complex community. But maybe in the field, they will be reacting differently. I hope I'm, yeah.
Thank you, Justine. Did you find any difference between the ones you soak and the other ones?
So we had some, so we didn't, we are waiting on this data. The one we soak, so what we did is we looked at the proxy. We only looked at, so the simplistic community, we looked at a lot of plants. On the data that I have now, we just look at 2 to 3 plants, just because we need a high number for the other part of the project. What we saw is when we took cane sample from the medium and the high complexity, we only took 2 plants randomly plated, and we find our bacteria back. So they were there. With the soaking, it was not as good. We find it only in 1. But this is something that we want to start, like try again, because we soak them only by putting them in. We could have just immersed them fully first, and they will have covered more the area of the vine when it was growing, on the cane when it was growing. So it's something that we want to retry.
Yep, thanks. And do you think this lack of microbiome or increase on GDTs is correlated to soil health? Or any of you or your team think it could be correlated to soil health?
Do you mean that they are maybe coming from the soil itself? Or
Yeah.
Yeah, so it's because we don't really know where they're coming from. We know that they are deep in the trunk because it's where we isolated them from. We suspect that they might come from the soil randomly or somewhere else. We know that, so I just show the 6 months result here, but we also also looked at 3 months post-infiltration of the vacuum pump one, 10 out of 10, and she found actually 1 of the Pseudomonas in the roots of the plant. But it was not present after 6 months because we, by then, have put them in a potting mix. So we think they might have moved into the vine through the roots. It's a possibility, but we don't know.
What's the stability or age or longevity of the microbiome? So if you were to be successful and inoculate nursery vines,
Yeah.
is it still going to be there in 10 years' time?
Very good question. I can't say yes or no, but I can say what we've seen before in the microbiome. So what happens is often young plants have a more diverse microbiome. So a lot of different bacteria at different level, like lower level. And once the plant mature, usually you get more stable with a less main actor microbiome. So hopefully by seeing what we see now at 6 months, and they are really abundant, we're hoping that this will be how we go to the mature microbiome. Because we don't really let them too much chance. We're just putting quite a lot of quantity in them without killing them.
Hello, Justine.
Hello.
The stability, the functionality, that's the cutting edge, isn't it?
Yes. Yeah. Yeah. I think until we have year 3 data, it's hard to know. How does it work? Does it work? How does it work? For the moment, we are trying to just, like at this point in time, we're just making sure that what we think will be important are there.
Because obviously we were restricted by what we had that was culturable.
Exactly. This is
And
good.
also we found what was in a mature vine that didn't have disease.
Yes.
We don't know if there was a succession from when it was planted to where it got there.
Exactly. Yeah. No, I totally agree. And this is true. Like, one of the we can have a lot of data, but one of the limitations, and number one limitation, is we need to have isolated the microbes that we think are important.
Any other questions for Justine? Great. All right. Thank you very much, Justine. Doesn't matter. All right. Lucky last. So probably a familiar face to most of you in the room, but our last speaker is going to present an update on the MB program, supported by UC Vision, called Predicting the Unseen, looking at new imaging techniques to understand and begin to predict yield. So thanks very much, Damian.
Yep, thanks, Abby. Firstly, a disclaimer, because I'm not leading this program, but we do have a pretty big team of people who are. Unfortunately, this video has lots of moving pictures, so I hopefully won't have to say too much, and hopefully won't get too many tricky questions at the end. So the program is a 5-year program. It started in 2023. Quite a big amount of money. Well, sounds like a big amount of money. It's $7 million in total, with 4 institutes involved. So led by Richard Green from UC Vision, and as part of the University of Canterbury, supported by Lincoln University and ourselves, the Bioeconomy Science Institute. So the program really is around the development of a digital twin, and notionally the idea, and I guess the business case for the proposal, was around predicting yield in vineyards. But essentially, the problem of plants and trees kind of growing through the season, producing lots of leaves and then hiding things that you might want to see, is a really wicked problem, I guess, in terms of agri-tech generally. And so the reason the government funded this program is to try and develop technologies that help overcome this occlusion and hiding of fruit and things of interest within the canopy disease, so that it can help facilitate the development of agri-tech solutions in the future. So essentially the program is it has a physical ecosystem part where 3D imaging and trying to recognize and understand what's in those images. That work's being done by the University of Canterbury. There's a set of data collection that's undertaken by Lincoln University and ourselves around ground truthing and actually observing and providing reference data. So this is kind of what we call the physical ecosystem. It's a really challenging ecosystem to be doing this kind of high-tech stuff within, as you'd be aware. We've got biological and seasonal variation. The plant architecture keeps moving throughout the season, and it's a very complex plant architecture. We overlay management and pruning systems on top of that, trellising. There are lots of things, wires, tendrils, that make all the imaging and resolving those images tricky. And field environments are harsh, and wind is a factor. Lots of things make this program difficult. So that's why the government's kind of funding this rather than the industry, because it's very risky stuff. So there's this kind of physical ecosystem side, and then in parallel we're running a digital ecosystem which includes 3D modeling. that Junqi, I'm not sure if Junqi is here now. Oh, yes, she is, yeah. That Junqi is leading. And from that modelling, we go into scenario prediction. And so this is really an iterative kind of loop that we're running in the program. But the benefits, ultimately, we're looking for more accurate and earlier yield estimation in vineyards, but as a tool to service other sectors as well, because this really, fundamentally, we're looking for a high-tech platform to serve agri-tech in general, because robotics and anything automated needs to be able to see and see very accurately. So this program has set itself some stretchy goals around trying to resolve imaging at a resolution and in 3 dimensions, sub a millimetre. So we're talking wires and tendrils and very small inflorescences. But we want the yields prediction to be early, so we've got a longer window to act. But also the digital ecosystem, we can really apply data-driven science and run simulations to test lots of different scenarios and hopefully accelerate the time to value. We'll start with a little film. Hopefully this is going to run. So this is a 3D reconstruction, so it's not a video. So you can delve into these images and run and look deeply into them. Also, Oliver Batchelor, who leads this work, he's kind of extracted the leaves out of the image so you can see the bunches, which is pretty amazing, I reckon. You can even see the petioles and what's the residual, if you like. And they're also sizing individual berries within that population. So this is kind of hot off the press. I only got this video yesterday afternoon, so and it is a lot of work to get to this point. Yes, so that's showing basically a berry size distribution within, and all those different colours represented kind of different sized berries. So yeah, so as I said, this imaging and image reconstruction, image recognition work is being carried out by UC Vision. And they've basically built a series of scanners over the first 3 years of the program. Each scanner be getting more and more sophisticated, higher and higher resolution. So the 3rd generation scanner now, I think, has 12, 16 megabit pixel cameras on it. So it generates a huge amount of data, which produces very, very high quality images. But the processing and the image processing pipeline is tricky and very time consuming at the moment. It's very data hungry as well. Plus we have, and Julian might be able to, if there are any questions, we do strike lots of kind of challenges in the field just with GPS and positioning and all sorts of things that you would expect, I guess, in trying to run robots and automated systems. At the moment, it needs to distance very precisely from the canopy is one of the challenges as well, and it needs very precise positioning to be able to stitch together both sides of a row coming the opposite way, if you like, back down the road. But as we saw before, the scans are amazingly detailed, and basically the segmentation of the images can give some pretty interesting and useful information. This is berry size, or this count of berries at different size distributions within the crop. And that ultimately, I think, will allow us to do some really interesting things from a research perspective, where we can follow the growth of individual berries, within a canopy as part of our whole yield assessment perspective, because we kind of, there's always a missing piece in terms of bunch growth and berry growth in terms of our yield estimations, and we think that's probably related to some proportion of the population growing at a different rate or not growing, that's influencing our final yield prediction. So this type of technology will allow us to hopefully resolve some of those questions. The second part of the program is around sort of the data validation, the ground truthing, and as well some sort of finer level modelling. This work is being led by Lincoln University, supported by Bayesian statisticians from the University of Canterbury. And so, you know, looking at kind of the challenges and the opportunities associated with this, really to try and add detailed kind of functionality and accuracy around some of the key phenological stages, and particularly flowering is one that's been identified as needing a lot more kind of functional modelling and our ability to help predict what's happening during flowering. So yeah, this work's being done by the University of Canterbury. They're also using and going to be using the 3D imaging to take a much more sophisticated look at vine balance. So historically we've used kind of rather gross measurements like pruning weight and yield and the ratio of those two things to assess vine balance. Now there'll be an opportunity to look much more closely at what balance actually means in terms of different types of shoot distributions within a population, the ratio, the individual ratios of leaf area to fruit on shoots, and how they might perform and how they might grow. So that's kind of a watch this space. There's a PhD student, Sagde, who has initiated his work and will go for another couple of years under Amber and Olaf's guidance in this study. And the third part of the program is being led by Jin Chi for the Bioeconomy Science Institute. So previously Jin Chi developed a very sophisticated structural functional model called Grapevine XL, which he has published, and that model was built from an international collaboration across many different institutes. It uses a platform called GrowImp and integrates a whole lot of different modules that operate at kind of an organ or even a tissue level within a single plant. It has sophisticated light interception, that's ray tracing, gas exchange, water transport, fruit development, carbon allocation and carbon transport modules that all work together within the model. It has evolved from being Grapevine XL into what's now Fruit Crop XL, because for the last 4 years, I suppose, Junchi, Plant and Food and now the Bioeconomy Science Institute, we've had a program of work that's been focusing around apples. So a lot of the model development that's happened subsequent to Junchi's first publications in 2018 and 19, a lot of the developments that's happened subsequently has been on apples rather than grapes. But we are in a position now with this program to bring those advancements that were made in apples back into grapes, which is what we want to do. So this video, I think, should run. So this is a simulation of apple trees growing in a FOPS system, which is quite similar to grapevines and extremely similar to a next-gen vineyard system. So hopefully you can see the fruit just starting to form there in the simulation. So it's running a full season, colour change, fruit ripening, and yeah, basically the model is able to do lots and lots of different things. So it can run from a single organ up to a vineyard kind of scale, so many different plants. And the simulations can be run from an hour through to a whole season. It is computationally very kind of expensive, very heavy. So the long simulations take a long time, but it is very powerful. And our aim here within this program is to import, I guess, 3D images from the scanners and basically calibrate the model to grow to be the same plant, the same digital twin, if you like, as the physical vines that have been scanned. So we're running the model is basically then creating the predictions and the physiology that's happening alongside what the real world imaging is giving us. So, and obviously we can simulate different growing systems, orchard layouts and crop loads, and also fruit growth parameters under different crops and water stresses. And yeah, as you saw in the simulation, it can give dynamic growth right throughout the whole season. This is just an example of what the model can do in terms of output. So this is water flux from trees, but you know, looking at the blue dots are observed values, and then we've got the adjusted predicted flux from the model, and the purple line is kind of the initial outputs of the model, and then once the model is calibrated, we get those kind of adjusted green lines. So very, very close to what's actually observed in terms of transpiration rate, for example. Junchi would be much better to speak to these graphs than I am, but these are basically three-dimensional representations of different internode positions on an apple tree, and basically the model is able to give the stem water potentials at a certain time of day across the whole tree. And leading on from that is a sugar concentration at the internode level, and likewise that gives you a fruit sugar concentration once the model has run a certain time, and basically can produce a distribution like that across the tree. So it's very, very powerful kind of modelling. Alongside that, we are also running, and we have a number of our collaborators here, so thank you very much. We are also running in-field kind of data collection while we're waiting for the quality of the imaging to kind of come through in the program from the University of Canterbury. We've been developing methodologies that simulate machine vision and then running those through not Junchi's fruit crop Excel model initially, but some simpler models to develop predictions. So these yield predictions we've developed from the program, we produce those by late December, so not long after flowering, and we've kind of come to the conclusion, we kind of already knew it, but we've satisfied ourselves that we really need to ground truth the inflorescence number, like the bud number, the shoot number, the inflorescence number. We really need to ground truth those things in each block each year, because they vary so much from a management perspective and also from, yes, kind of springtime noise, and that, you know, that might be grass grub, it might be frost, it might be all sorts of things. wetters. So we really need to have some level of ground truthing at a block level each season. But from there, our modelling is doing okay for the moment, but it does, so kind of 60, we'd set ourselves an aim of plus or minus 15% by mid-season, so by the end of December. Our modelling at the moment, kind of 60% of our predictions hit that target, but we do have a tendency to overestimate. And for the most part, that overestimation happens in vineyards where probably you'd assess that the kind of individual vine vigor, the soils are poorer, there are other things kind of happening. And so we're overshooting mostly on lower vigor sites. And so our future focus on, if you like, what's causing or what's driving our overestimate from a yield perspective, we're focusing in on why do those vineyards in fact not achieve the yield that they should, or we think they should. But there's some very complex kind of interannual and a lot of kind of non-climatic factors happening in their soil and rootstock and all sorts of other things. So we need to tease that out a little bit. But that's kind of where we're at, I think, in terms of the program. We're sort of halfway through, so progressing, I think, pretty well. Yeah, a few take-home measurements and messages. So the scanning resolution has vastly improved. Field capture is still challenging and always will be. And I think at our last workshop, you know, for the most part, I think for the next probably 5 to 10 years, we will mainly be looking at these 3D imaging techniques as a research tool or as a tool for companies and viticulturists to gather reference information. It's not going to be the kind of technology that we can broadly apply across every vine in a vineyard. But I don't think that necessarily matters if the accuracy and the value of the information that we can collect is there on, you know, 20 vines or 40 vines or whatever it might be. The image recognition and processing pipeline is certainly making great strides. It has in the last 6 months. But yeah, as I said before, we can run models, but we need ground truthing each year. So we need good quality imaging for that. We tend to overpredict, and we want to focus on why that is. And we really want to hone in on the physiological drivers of what's I guess constraining yield in those situations. So I won't go through all of these, but yeah, acknowledgements particularly to MB and for funding this program, and certainly the leadership and the vision, excuse the pun, that UC Vision, Richard Green and his team have had. So thank you. Happy to take some questions, yep. Did I go a bit fast?
Yeah, modeling has a way of doing that. Junchi, do you want to set the record straight on anything? No.
Maybe not a question, but just an additional comment that as we are heavily reliant on crop estimate, yield estimates right through the season, probably more than ever now than what we've ever been, it's great to see strides of progress into the new technology, but at the same time having a system alongside that working that is still valuable and useful for us as grape growers in the industry. And we shouldn't probably steer ourselves blind that in time to come, all that sort of thing that we're talking about here, the high-tech stuff, I'll probably won't see it. But look, in the meantime, I think we're pretty much on the ball. And yes, there are some outliers either way, and we'll just have to work around it.
Yeah, and I mean, this actually relates to a conversation you and I had last week. But, we do things pretty well here, and I think relative to other regions and other countries, we have great quality information. We have VineFacts publication, and we already publish at a regional level and sort of to a point at a sub-regional level yield forecasting, and the statistical models that we've had, to make those forecasts, they've been great and they're still valuable. And so we have those as a kind of a regional overlay. But what we're talking about here is really getting down to block level predictions. And so that's, there are obviously a lot more variables that can kind of come into play. But the idea is really just to build up layers and be able to kind of produce forecasts at smaller and smaller scale ultimately. But not forgetting that we have these kind of regional forecasts and a number of producers are already running kind of their own models using their own historical data to help support. So, and that's kind of the thing as well that has become quite clear in this program, is that introducing historical data from individual blocks will be hugely valuable in terms of the ability to predict what will happen within those blocks. So that's an area we would like to kind of, and working with Brigato, hopefully be able to increase our kind of our big data approach to these things. And even when this program started, we were talking a little bit about AI, but there are huge opportunities as well for AI to kind of come into this space and really start to help us. Once we can generate extremely kind of detailed information, we can then throw AI at it to start to try and unpick what's going on. So, yeah.
Actually,
Sorry.
I did. It was really more about you said your overestimation of yield was from lower vigor blocks. Have you pinpointed, is it the berry, because presumably you'd be able to see more of the fruit in those blocks. So your visual determination of berry size or whatever should be more robust.
Well, we're doing it pre-flowering. So we can see the inflorescence number, but everything from there we're running just as a model prediction.
Gotcha. So December is purely modeled based on...
Correct, yeah.
So you're not looking at berry number?
No. Well, the model is generating berry number, but we're not validating that yet because we haven't got the easy way to validate it.
Yeah, gotcha.
But certainly kind of individual shoot vigor is playing a part, and we need to look more closely at kind of inflorescence size, not just number, but inflorescence size and then obviously fruit set. There's a whole bunch of things that we want to look into more closely. Yeah.
Thank you.
Mm-hmm. Thank you. I think it's a bit more, I'm thinking, you're talking about prediction, but I'm thinking down the line, if our project, we're showing it's working and we're planting some plant in the field with different microbial community versus control plant, could this technology help us monitoring how the plants, because we will need to go down this path after?
Absolutely, and in terms of phenotyping for breeding programs and the like, it is ready made. For that as well, yeah.
Perfect. Thanks.
Any last questions to round out our session? If not, please join me in thanking Damian and Justine and Michelle. I just want to briefly finish with a couple of acknowledgements. The first is to Stuart Field, who is normally up here having the job that I have. He's been instrumental in getting these sessions organized and keeping them running, so a big thanks to NMIT and to Stu. Also, the ability to archive these sessions and have them to access back to and share with your colleagues, I hope is going to be something that's really valuable, and that's something that wouldn't have happened without MRC's support. So thank you for that. And then just lastly, just for making the time to come out. The recordings are not so that you don't come in person. Please continue to do so. I think the New Zealand wine industry is in a period of change, and it's easy to innovate when everything's going well. It gets challenging to innovate when things are actually the rubber's hitting the road. It is all the more important that the dialogue between the science community, which many of you probably also know is in a period of flux, that we keep those lines of communication really open. It could be a point of difference for us, continue to be a point of difference for us going forward. So thank you very much for carving the time to be here, both our presenters, two of whom who came from out of region, and all of the industry folks in the audience. We really appreciate you. Cheers.